All cells are bordered by phospholipids, which we learned about in the previous chapter. Recall they are both hydrophobic and hydrophilic. They have polar heads, and those polar heads are hydrophilic, and nonpolar tails, and those nonpolar tails are hydrophobic. That's what enables a bilayer to form. In the bilayer of a cell, we have proteins. And the proteins that are placed within the bilayer of the cell have a variety of functions. And if we look at this slide, we can see recognition proteins over to the left. And basically think of these as kind of like the fingerprints of the cell. They identify the cell and enable the cell to identify other structures. The one labeled C is a receptor protein, which does just what it sounds like it does. It enables the cell to receive substances, to bond with other substances, and then from that bonding perform further actions. D is a transport protein, and a transport protein in many cases is transmembranal. As you can see, it's going all the way through the bilayer, and that enables transportation of substances both in and out of the cell. And then E is an image showing us a transport protein that also has an enzymatic component to it. So this is a transport protein that has ATP synthase, that as hydrogen ions move, ATP is produced. So it's not just the phospholipid bilayer that makes up our cell membrane, but proteins as well. This is a generalized view of a phospholipid bilayer. This would be an example of what a spherical cell would look like, and the fluid would be the cytoplasm where all the organelles of the cell are contained. So what are some of the components of eukaryotic cells? Here's a nice table that summarizes them for you. I'm going to discuss them as I show you some images of them. So here we see an image of a nucleus. This nucleus is bounded by a nuclear envelope, and inside the nucleus we have genetic material, that's the DNA, as well as the nucleolus. That's a concentrated area with ribosomal RNA, which will be very visible in cells that are actively undergoing protein synthesis because they need ribosomes enabled to do that. The nuclear pore is just as it sounds. It's basically a pore or a hole or passageway that allows substances out of the nucleus. The nucleoplasm is, in essence, the cytoplasm of the nucleus. And notice external to the nucleus, we have an endoplasmic reticulum. In this image, it's the ER. And it's an endomembranal structure. It's an organelle inside of the cell that is responsible for packaging in moving substances through not only it, but other constituents of the endomembranal system. If it has dots on its surface, as you can see here, it is known as the rough endoplasmic reticulum. And what happens is ribosomes in the cytoplasm attach themselves to the endoplasmic reticulum, and as the polypeptides are being made from the ribosomes, they travel through the rough endoplasmic reticulum and then usually through other pieces or parts of the endomembranal system to be further processed. So to summarize, part of the endomembranal system is the rough ER, which has ribosomes attached to its surface, which enables polypeptides or proteins to be made. And as those polypeptides or proteins are made, they are fed through the rough ER and continue into the smooth ER, where they can be modified. The smooth ER can also make lipids, decompose or break down through hydrolytic reactions, carbohydrates and fats. And the smooth ER is found in large quantities in organs, which are responsible for detoxification, such as like your liver. These materials can then pass into the Golgi body or the Golgi apparatus. And the Golgi apparatus is the organelle that is involved with packaging and distributing cellular contents. So it's kind of like the UPS store It finishes up any additional molecules that need to be added to what enters it, sorts it, tags it, and ships it in vesicles that then fuse with the plasma membrane and basically are shipped outside of the cell. We also see as part of this system lysosomes, and lysosomes contain hydrolytic or digestive enzymes that are responsible for decomposing or breaking down, or in essence digesting cellular contents that could be just worn out cellular parts, or they could be substances that the cell has brought in and need to be broken down. So here we see a more detailed view of that pathway. So from the nucleus, from our DNA through the process of transcription, we make RNA. The RNA leaves the nucleus and goes into the cytoplasm. And in the cytoplasm, we have ribosomes. Some of those ribosomes are associated with the endoplasmic reticulum. So the messenger RNA will be read by the ribosomes and translated into proteins or polypeptides. Once they are produced, as you can see kind of in the middle upper corner right here, you can see this polypeptide being produced from the ribosomes on the endoplasmic reticulum membrane. They will then be contained within these phospholipid bilayer bubbles called vesicles. And the vesicles bud from the ER, and as they bud from the ER, they move through the cytoplasm, and some of them move directly out of the cell. Others can move and join the smooth ER, and still others can move to the Golgi apparatus. So here's an example of the vesicle moving to the smooth ER. So in this example, our green threaded polypeptide or protein here was made from ribosomes on the rough ER, and now it's been transported to the smooth ER, and it might be modified, lipids added to it, or other molecules. And as it leaves the smooth ER, it's going to go to the Golgi apparatus or the Golgi bodies. We have step 4, the Golgi body, and in this organelle, our polypeptide is going to enter the Golgi body, and as it arrives from the endoplasmic reticulum, it will be modified, sorted, and shipped outside of the cell, and it leaves the cell by way of the plasma membrane. And you can see at step 5, an internal vesicle that has budded off of the Golgi body, merging with the plasma membrane. And that green polypeptide is being dumped externally outside of the cell.